As I mentioned earlier, all cells have a plasma membrane, and that's what we see here in this diagram. Plasma membrane has its own chapter that we'll be looking at soon, but it is composed of phospholipids, which are a phosphate group on one end with a lipid tail on one end. The phospholipids like water, so they're said to be hydrophilic. So there's a hydrophilic region here and here, while those lipid tails do not care for water. They're hydrophobic. They're repelled by water. And then there are lots and lots of proteins and carbohydrates and other things stuck into that membrane. And so every single living thing has that membrane. And some of these proteins act as tunnels to let things through, or act as enzymes to run chemical reactions, like the protein stuck into the membrane of a mitochondria, for example. So we'll talk much more about those soon. Before I go on, I want to point out that we mentioned the structures found in prokaryotes. Most prokaryotes are like 10 times or so smaller than a eukaryote. They're very, very small, which means they have a very high surface area to volume ratio. And surface area to volume ratios are an incredibly important thing to understand when it comes to biology, especially as you learn about structure and function and physiology. Just to give you an idea, so here's a box. So the lines I've drawn, this is the edge of the box. That is the surface area. And inside is the volume. And so when we're talking about surface area to volume ratio, it's literally SA over V. And that's the ratio we're talking about. And think of it like a city. You have a bunch of buildings, a bunch of people living in the city, and you have, I guess my horizon's a little crooked there. You have one road going into that city, right? One road going into that city, and that's the only way in and out. Now, the surface area is the membrane. That's how much stuff can get in and out of the cell. This is like the highway to get in and out, is the membrane. And so if there's a whole bunch of people, so volume, a whole bunch of people in the city, you're going to need more than one road, right? So the relationship of edge to volume is really important, because surface area is basically a measurement of how much stuff you can get, like how much oxygen and food you can get into the cell, and how much waste you can remove, how much you can get. And volume is a measure of how much you need. And really small cells have a higher surface area to volume ratio than big cells. Big cells have a hard time surviving because volume increases faster than surface area. So when you get huge, you have a lot of need, but you have less highway to get things in, okay? And you can see that here. In this example, we have this little cube, and each side is 1 unit big, right? So the surface area for that unit, if you follow the formula here, is 6, okay? The total volume for this thing, this same box, is 1. And so the ratio here is 6. But if you make this box 5 units big, suddenly its surface area is 150. So it's a huge surface area, right? And its volume is 125, which is also huge, right? It has a big surface area. It has a big volume. But if you put surface area over volume, the ratio is smaller. So how do we make a big organism that can survive and get enough nutrients to all of its cells? We make it out of a whole bunch of tiny cells. So that blue whale is made up of trillions upon trillions of tiny cells that still maintain, each cell has a high surface area to volume ratio. That's why cells are small. But even eukaryotes have cells like 30 to 50 microns in diameter. So think about 50 microns diameter average maybe. That's kind of big for a cell. So they have to have lots of rooms inside. I mentioned a prokaryote is like a one-room cabin. Eukaryotes are like a house with lots of rooms, and different chemical reactions take place in different rooms. And all of that extra membrane inside gives them a little more surface area. So here's an animal cell showing all kinds of things going on in there. More than I can point out in one go. But this is a great figure to come back to and review. You'll see a lot of things you recognize. Here's the nucleolus. The nucleolus makes ribosomes, right? And there's chromatin in there. You know what that is, right? It's DNA, DNA and protein. The nuclear envelope is a double membrane that surrounds the nucleus. This shows you all of these things interacting here. And then the next figure in your book is a plant cell. And they don't tell you as much stuff in the plant cell because they only label things that are unique to plants for the most part in this one. They still label the nucleus, but they don't have, they label everything, but they don't have explanations for everything that you already saw in the previous slide. And notice there are some things in the plant cell that you didn't see in the animal cell, and vice versa. And I'd like you to pay attention to that and figure out what some of those are. So what do plant and animal cells have in common? What is unique about each? I'm going to let you make a list of that, and I will make sure to put a couple questions on that on your test. But I will say one thing that they both have in common is mitochondria. I'm going to tell you that one because students often think that plants don't have them, but plants do have them. They make sugar so they can eat it with their mitochondria, right? They can break it down with mitochondria. So that's something that I just wanted to point out. All right, so what I'm going to do then is I am going to lecture then about the endomembrane system. So the endomembrane system is all of the membrane inside of the cell. Maybe not all of it, but all of the membrane inside of the cell that's kind of connected to each other, either connected directly or through transfer or transport vesicles, the taxis inside of the cell that transport things. They're connected in some way. So the major things I'm going to mention, and I want you to read more on these because I'm not going to hit them all real hard. I'm going to introduce them. The nuclear envelope, the endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, and of course plasma membrane will have its own chapter. So those are the parts of the endomembrane system I want to focus on. And I'm going to start on the first one and probably break this up into a couple videos. So the first one I want to mention, is the one I just rocketed past. Okay, is this one right here. This is the nuclear envelope, okay? And you can see it written down right there. And it's a double membrane. It has two membranes. One encircling the other. And then there are holes in it called pores, nuclear pores. And from these pores, this is how RNA gets out of there, right? Because remember, transcription translation, if you've learned about that already, takes place inside of here. So if you don't know that, RNA is made in here, like messenger RNA, and then it comes out of the nuclear pore, and then it goes to a ribosome where protein is made. Okay, so something that you should be aware of. So here's a close-up of a nuclear pore and some ribosomes kind of stuck right here waiting to get to work, right? Inside of here is also chromatin, which is DNA all wrapped up around proteins. Now, attached to the nuclear envelope is the rough endoplasmic reticulum. The rough endoplasmic reticulum is a series of membranes all folded up that is responsible. One of its primary things is protein production, and it does that because it has a whole bunch of ribosomes embedded to it. These are said to be bound ribosomes. Now those ribosomes can come back off again, of course. You can read about that in parts of your book, but they're attached to it like rhinestones. They're stuck all over here, and there's some stuck to the nuclear envelope as well here. But messenger RNA would come out of here, attached to a ribosome, and it would make proteins, and those proteins would be made into the rough ER. So just look at the ER real quick. Here's the nuclear envelope again. Here's the rough ER, and then there's a structure known as smooth ER, which is a little different shape and has no ribosomes on it. The rough ER makes protein, and it's also responsible for some of the extra membrane that's going to be made by the cell. And the smooth ER is going to be involved in making carbohydrates, making lipids, like hormones. Like if you have a gonad, like an ovary, it's going to have tons of smooth ER because and probably rough ER because it's making lots of hormones, which are made of lipids, right? That's going to be there. Another thing that the ER does in general, They're often involved in detoxification, breaking down certain chemicals. So you'll see cells that are in the liver with a lot of ER as well. So the rough ER is going to be making a lot of protein and things. So what happens here is proteins are made or lipids are made here. So let's just draw, I'm going to draw a really crude ER. Here's a piece of the ER, and it makes something in here. And then this membrane is kind of like a soap bubble. Phospholipid membranes are kind of like a soap in a way. And this stuff can just come up to the edge and pinch off, and this membrane can pinch off and form a bubble. And this is called a vesicle. And this can be transported through the cell to some other place. And the next place it might go might be the Golgi apparatus, right? And it might go there and do something, be modified there, and then go off to do something else. So what I'd like you to do, of course, is review in your book the functions of the ER and of transport vesicles, and build on what I'm saying here and make a good set of notes. I may have you do a concept map on this for an assignment. But in the next video, we're going to pick up with some of the other parts of the endomembrane system. And then we're going to also move forward then and look at other parts of the cell that are not necessarily part of the endomembrane system itself.